Thanks for being here after a really nice evening. Survivors, yes. So today I'm going to talk about the origin of animal form. So I'm going to try to contribute, or we're going to try to contribute to answering a question that since the 19th century biologists are thinking about or trying to resolve, and is how the variety of body plans evolved from a common ancestor of all animals. So the idea is that a theoretical organism called Gastrea originated, and we thought at the beginning that originated here. So Metazoa is this pink square. And traditionally, we assume that Porifera branched earlier, and that's why they don't have gastrulation or tissue organization. But then, at this point, a gastrula-like organism evolved, and from here, a variation on this body plan and the genome regulatory networks originated the diversification of animals. So then we had animals like divided into groups, Parazoa and Eumetazoa, organisms with real tissues. But later, in 2023, a new paper came showing that Ctenophora, the phylum Ctenophora, was the basal clade of Metazoa, and Ctenophora gastrulates. So that implies that the gastrula originated here, and that Porifera lost the gastrulation capacity and the organization. And we see something similar here with Placozoa, that is a very simple organism in the, at the adult stage. And with this new finding, we get gastrula back to the root of animals, and we are kind of reviving an old idea postulated at the late 19th century by Haeckel, saying that Gastrea was the ancestral, something like Gastrea was the ancestral animal that gave rise at all other animals. So what's the big deal with gastrula, with the gastrula stage? So the gastrula has two distinct tissue types, endoderm and ectoderm in basal animals, which are Porifera, Cnidaria, Placozoa, and in Bilateria, it has 3 tissue layers. So these cells, or this structure originated from a blastula that start differentiating and organize 2 different cell populations with distinct chromatin structure and genetic profiles. So in a way, it's the first step on a differentiation cascade that is gonna end up forming specific phenotypes associated to tissues, and at the end is gonna end up forming animal forms. Yep. So, for example, from a dipoblastic ancestor, the origin of a mesoderm, an extra tissue layer, gave rise to the bilaterians and all the different body plans that we see. It allowed the formation of tissues like the digestive system and everything that is derived from the mesoderm. Other thing that happened when we experience, or when the gastrula experience modification, is the change of axis. So within Bilateria, a change of axis originated the deuterostomates. So instead of having a blastopore that is going to originate a mouth in this group, and something changed, and this blastoderm originated the anus, giving rise to the deuterostomia, a group that allowed the diversification of animals, again, including vertebrates and echinoderms. So studying diversification and selection at these stages is important to understand how body plans emerge in a big scale. Okay, so if the gastrula is a simplesiomorphy of all animals, one may think that the process and the mechanisms regulating gastrulation should be very conserved. But when we look at gastrulation in the animal kingdom, we see that the mechanisms are very diverse. So gastrulation occurs in many different ways, and there is not phylogenetic signal attached to it. So we cannot reconstruct animal lineages just by looking at the way that they gastrulate. So that generates like a conundrum, like what's happening here? So if the morphology is so conserved, why the way cells regulate gastrulation are so diverse? And that idea make us rethink the way we talk about development. So from the mid-19th century to nearly the end of the 20th century, we thought that development was immune to selection, so that the animal form was only selected and diversified in later stages of development and in adult body plans. But once we start seeing a lot of variation, also at early development, we identified two possible ways, two possible places where natural selection could start generating adaptations and generating diversification. And then, to make things a little bit more interesting, in 2016, there is a paper, and the authors say, hey guys, let's stop thinking about genes. You know, everything is so gene-centered, and let's try to stop thinking about gene determinism. What if genes don't have, so he said, random mutations and natural selection by itself is not able to explain the origin of complex structures like limbs or organs. So in this paper, they start postulating the idea that there are mechanical forces and geometrical constraints that happen early during development, especially in the blastula stage, that define body plans or what the body is going to be. So to some stage is the constraint of cell division within membranes that is going to start restricting animal morphologies. But the genome and gene expression is also involved in directing cell differentiation. So it's a two-way process where genes have something to do, but mechanical and spatial and geometry of cell division is also shaping the process. So as a biologist doing genomics and comparing genes and looking in genes and protein domains, I would say, well, but so what about developmental signaling pathways? So there are a few developmental signaling pathways, and they all well conserved through all metazoa. So what's happening then? So for example, the Wnt signaling pathway, the BMP, FGF, Notch, well, they determine axial polarity, cell differentiation. But again, these pathways act within the range that is created by the divisions of the cells early in development. And if we look only at the gene function, at the protein structure by themselves, we could go to probably wrong conclusions, because I think it's the interaction between gene products and the environment that end up shaping animal evolution. So I would like to think of gene networks as a evolutionary unit, and should focus on understanding how these evolutionary units or transcriptional networks are being selected by the environment. Okay, so to answer the question of how diverse is really the transcription profiles involved in gastrulation, and how conserved, there's a conserved aspect, there's also some divergence. We wanted to understand how concept was the mechanisms involving gastrulation, how diverse they were, and we used a model, the coral Acropora. It's a Scleractinian coral from the basal, the most basal here. So this is Bilateria, this is Cnidaria. Cnidaria is considered the sister group of Bilateria, so studying processes here will allow us to understand what was here, what was the last common ancestor between Bilateria and Cnidaria, and corals are here in Hexacoralia, the base, the more basal class of the Cnidarian phylum. So the group is becoming a really interesting model to study developmental mechanisms. So here we are, one of the genus. This is a genus, and we're working with two species, Acropora tenuis and Acropora digitifera, and the species of the same genus diverged 50 million years ago. So that's a long, long time for a genus for two species to have split. Basically, the early development is pretty the same. The corals spawn, the gametes mix in the water column, fertilization takes place in the plankton, and early development occurs in there. And they spawn at the same night every year. So the ecological conditions are pretty much the same. But we have a long time of divergence. And we need to take into account what's happening to genomes with the time, what time does to genomes. And we see that in some loci, Acropora digitifera experience duplication, gene duplication, tandem repeat duplication. And there's an idea that after following gene duplication, we could get to neofunctionalization or a specialization of a function. Yep. So we wanted to see how gene duplication events could shape or could change the transcriptional networks involved in gastrulation of those two coral species. We use a pretty straightforward transcriptomic methodology. We work with 3 stages, blastula, gastrula, sphere, that is a postgastrula, it's like the initial of the larval phase. We ensemble the transcriptomes with the reference genomes, or find orthologs with the ortho-MCL, and construct co-expression networks and visualize them using cytoscape. And this is the life cycle of the coral. So this is the blastula. We have 3 replicas of this. We have the gastrula, the process when the invagination, or when it's not invagination, when this stage is like a brown sheet. It's like a flat layer thing, and this thing starts doing like this, closing like this, and this is going to be the blastopore. And well, that process is gastrulation. Is the one that we have here, and we have the sphere when cell differentiation is occurring. We have mobility. This is like an early larval stage. The animal is motile here, so we are comparing transcription profiles between these 3 species per species, and we collect them at particular time points, and the morphology looks exactly the same between the two species. So after we sequenced, we found that the digitifera, we were able to ensample more transcripts than tenuis. Maybe it's because the genome is bigger, there are more events of gene duplication, so therefore more transcripts, and something that started kind of showing us or making us think is that here we reconstruct replication, but we see that gastrula and blastula are closer together to each other, so like in an embryonic cluster, than the larval stage. But for the other species, we see that gastrula and the larval stage are closer together, and PC is the distinct stage. So we start seeing different patterns of gene expression. What we did next was to find orthologs, and we find, I cannot read the number here, but from that number, I think 7,000 ortholog groups, and from that, 86% had a one-to-one ortholog relationship. So, well, yes, it's the same genus, they have pretty similar function. It makes sense that the percentage was this high. And what we did next was to compare, to find differentially expressed genes. So we did pairwise comparison, and we did also chemical clustering to detect wider expression profiles, and we found conserved modules in both species, but the modules are conserved, but they are not expressed at the same time. So we have, this is what is expressed in gastrula, but we have a submodule here that is expressed also in the other gastrula. So we can call this the core gastrula transcription profile, and we find that some other modules are expressed in a different stage. So this is called heterochrony. So it means the same model, the same genes are expressed, but in a different temporal pattern. And we validate the same when we did the analysis with the chemical clustering. So then we took the genes that were shared, that were DEX in both species, and did enrichment to find biological processes. And we found here in the intersection things like wind signaling, gastrulation, yes, so snail, brachyury, molecules known to be involved in gastrulation. So there is a conserved aspect of gastrulation, but not necessarily is expressed at the same time. What we see with the DEX that are one-to-one orthologs, but are only differentially expressing what species, is there are some of the same processes. So it suggests some redundancy in the networks that are being overexpressed in one species over the other. What's the meaning of that? No idea, but there is there. So basically, we are finding variation in early development, which is consistent with the hourglass model. And then we focus in groups that are specific for each species. In this case, the imparalogs. We found that many more imparalogs were differentially expressed in Digitifera gastrulation. And the interesting thing here was that most of them, we look at a few, we haven't looked at the whole data set, they have different expression patterns. So one of them does something, the other one, they are doing different things. But the Acropora tennis parallels that were less, they kind of doing the same, or particular, yes, pretty much similar expression patterns. So there is variation there during early development. What does it mean? How does it work? How are they selected? No idea, and that's part of the question. And when we look at correlation networks of expression, we see that the imparalogs create independent modules that interact with orthologous genes that are kind of hub or key for the processes, but is doing is creating an alternative network of regulation. So is it robustness? What's the role of that network? No idea, but it's there. And the same with isoforms. Isoforms also specific for a particular species. They are also creating networks that are alternative network to the main one. So in summary, our results are consistent with the hourglass model. We identify species-specific gene expression during early development. We identify conserved models or genes involving gastrulation, but they were expressed at different times. We also found that the patterns of expression of imparalogs from one species and the other are different. In one species, the imparalogs do different things. In the other, tend to do the same. And imparalogs and isoforms that are specific for each species, they have the ability to form independent networks from the original one. And with that, I want to thank Juan Pablo. He was the one that did this. He did all the bioinformatics, my student, and my collaborator. He couldn't be here, from Universidad de Antioquia, Hector Alejandro Rodríguez-Cabal, and this work was done in Okinawa. Species are from the Indo-Pacific, and yes, that's it. Thank you very much.
Thank you very much, Alejandro. Any questions from the audience? Please.
Very nice work, Alejandro. What kind of thinking, do you know if the localization of these genes is kind of correlated between the species you analyze?
Yes, that's we wanted to do the genomics, but I didn't wanna. So we work on it. This is the first result. This is the first step. We have to do that. We have to look into genome architecture.
And if I can add a little second question,
Yeah, please.
there's also some kind of information on other nidarians like the Medusozoa, transcriptomic. Do you have time to look what happens with these genes in these other species as well?
We want to do that. There's a lot of data. We want to do that, but we don't have time and we don't have complete resources. It's difficult. But we're getting there.
Any other questions from the audience? I have a couple. Very nice work. How conservative were the non-coding RNAs that we identified among the two species?
That's a nice question. We are working with another professor, Alejandro Reyes-Bermudez from Universidad Nacional, and there's another master thesis that is being developed on that topic. And yes,
Because
no,
in general, they
yeah.
are not so well conserved
Yes.
when you take into account the primary sequence. But
Yeah, they're not concerned, but
the
the
secondary structure maybe could be so.
yes, and we did that. We compared secondary structure of long coding RNAs, and if you see here, see, 10% of the transcripts are non coding, so
Yeah,
they play a.
But you use CPC to identify.
Yes, only potential. Yes.
Only CPC.
Only CPC. Yes, so far only CPC.
I would say that to include more, because more tools, of course, you already have the work done, but the future works I would include more, let's say 3 or 4 at least, apart of using the BLASTX or Diamond against the other proteins that you have, because most of these tools they are developed taking into account the model organisms in the training. This is a bottleneck, but of course it works. I use it both tools and I integrate a little more just to avoid any false positives.
Yes.
And the last question: did you use any metrics to evaluate the module's conservation in the co-expression analysis? You have the co-expression analysis that you did, and you identify the module's conservation, right? Did you use any metrics to evaluate how conserved and how did you? Yeah, to hiciste el análisis de co-expresión, ¿cierto?
Sí.
Y después identificaste la conservación de los módulos. ¿Hay alguna métrica que usaste? ¿Cómo hiciste esa conservación?
No, básicamente lo hicimos por tiempos.
No, pero tú entre una especie y la otra, ¿cómo fue?
O sea, la conservación de la expresión
De los módulos.
de los módulos.
Fue la presencia y ausencia de
Por
genes
la presencia
autónomos.
ausencia de sí.
Porque hay unas métricas de biología que se pueden usar. Ahora no.
We'll write to
Beautiful work, very interesting work. Congratulations.
Thank you.
Thank you. Please go ahead.
I have another question. It's regarding the enrichment analysis. I know for a fact that the Nigerians have a known lack of GO annotations and Kega annotations. I don't know the case of the ones I work with hydroponics.
Yeah.
But I wanted to ask you, do you have problems with the enrichment analysis because of the lack of GO terms associated to the differential expressed genes? Or
No, like, if you see, like, pretty much, no, I think the core genomes are really well annotated. So most of them have both terms, and 76% of the core genes are orthodox to humans. So I really don't experience that problem.
okay, because with my genome, I only got from 54,000 genes, I only got 23,000 genes that got a GO term associated.
Yeah. So, but Scleractinian, they're weird. They are the divergent, so I don't know what's happening to the problem. Acropora is the basis.
Thank you very much, Alejandro.
Thank you.
An applause, please.